Yeah, so I'm a Professor of Translational Drug Discovery at the Medicines Discovery Institute. The Medicines Discovery Institute is not related to the Medicines Discovery Catapult. We are a separate organisation. And next slide, please. This is just a brief overview of who we are. We opened in March of last year. Investment from Cardiff University and the Welsh Government, about £40 million. And the directors are myself as the head of biology and Simon Ward as a medicinal chemist. Next slide, please. And this is our pipeline. So this is our existing pipeline of projects. Obviously, this has not been assembled in the last year. This is some projects that were started when we were at the University of Sussex prior to moving to Cardiff University. And so that just gives you the flavour of the types of things that we're working on. These are drug discovery projects in an academic setting, so we get serious amounts of funding from the Wellcome Trust and the MRC to conduct a preclinical drug discovery. And our goal is to identify novel molecules and then partner them with pharmaceutical companies or other partners that can then take them through the clinical development. Next slide, please. Oh, thank you. So probably best to define what we mean by low-hanging fruit. So the low-hanging fruit in drug discovery are drug targets with a high probability of success. So things that is worth investing in because they might well actually work in the clinic and get to the market. And that's criteria for that would be a drug that's safe and well tolerated. And obviously, you'd like to have a good chance of achieving efficacy in the clinic. So you like preclinical models to build the confidence, ideally a well-defined patient population, and meaningful clinical endpoints. So clinical endpoints that are perhaps a bit more sophisticated than the rating scales we currently have in psychiatry. And I guess ideally, and somewhat paradoxically, compounds where you have a mechanism already on the market, so you know that that mechanism is safe and well tolerated. But then again, you would then have to differentiate against that molecule. On the other hand, the high-hanging fruit, click on please, Laura. Next, please. Yeah, so these are the high-risk, high-reward targets. So the higher hanging fruit are things where very sexy targets, presumably sexy because we don't really know much about them. They're a novel mechanism. So you don't really know if they're going to be safe and well tolerated in man. And you ideally base those targets around an understanding of the disease mechanism. And in neurodegenerative disorders, we know much more about the disease process than we do in psychiatry. I think a good example of that would be the amyloid-related targets in Alzheimer's disease, where we understand the pathology, we understand the genetics of at least the familial forms of Alzheimer's disease, which all point to the amyloid pathway. And next slide, please. And so in terms of developing a new drug, there's the old-fashioned way, which is obviously no longer tenable, of serendipity, where you have a hypothesis and inject some animals with some drug and see what happens. But those nevertheless gave us the lithium from people like John Cade and Paul Janssen and benzodiazepine from Leonard Sternbach. And as Laura referred to earlier, that was the golden age of psychopharmacology in the late 50s and 1960s. And there's drug repositioning, so things like Viagra were identified initially and developed for high blood pressure, and then a somewhat obvious side effect appeared in clinical trials, and that drug got repositioned. The other possibility is to take an existing drug and make it better. So a drug with side effects, if we could dial out the side effects, then that still have a significant patient impact. And then perhaps the sexiest and the highest hanging fruit are hypothesis-driven drug discovery, where you understand the disease pathways and the proteins involved, and therefore which proteins to target. And again, the example of amyloid pathways in Alzheimer's disease is a really good example. But then it's a good example as well how even understanding disease mechanisms does not guarantee a successful clinical outcome and a drug on the market. Next, please. So this is our portfolio again. So I've tried to rank them. Could you click on, please, Laura? Next, please. Yeah, so this is how I've exemplified our projects that we have in the portfolio. We have what I call a low-hanging fruit, which will be a GABA receptor alpha-2/3 PAM project. I'll talk briefly about that a bit later. An AMPA receptor PAM, GABA NAM project, medium height of the fruit, if you will. Limb kinase project for Fragile X. That's a high-hanging fruit. It's an interesting potential mechanism, but really does need to be validated with some good compounds. Now we've got some earlier projects which are, by extending the metaphor perhaps a shade too far, these projects are yet to be pollinated and develop into fruit. Next, please. I think a really important distinction is between neurodegeneration and psychiatry drug discovery. So the panel on the right-hand side is a really simple but very illustrative cartoon from a review from Menipangalos in 2007, looking at cognitive decline, presumably in Alzheimer's disease, and differentiating between symptomatic treatment and disease modification. So in neurodegenerative disorders, we have relatively well-defined pathology, and we also have familial forms of disease. And so we can actually envisage developing disease-modifying strategies along the lines as we've seen for the amyloid hypothesis in Alzheimer's disease. And then there's the symptomatic treatment. So symptomatic treatments in neurodegenerative disorders have played second fiddle over the last decade or so to disease-modifying approaches, but they still have a very important part to play. And the idea of symptomatic treatment is you can alleviate the symptoms, but when you stop the treatment, as shown in the red line, you revert back to the underlying disease course. Next, please. So psychiatric disorders, in contrast, the disease process are poorly defined in comparison to neurodegenerative disorders. So we have multiple small risk factors emerging from the GWAS studies. And understanding the basic underlying disease causes has prevented us from going for the disease-modifying approaches to psychiatric disorders and restricting us to symptomatic approaches. But then there's nothing inherently wrong with a symptomatic approach to psychiatric disorders from the patient point of view. Next slide, please. Yeah, this just echoes some of the comments made by Laura earlier. There's been a decreased investment in pharmaceutical companies in neuroscience drug discovery, and what's been left has primarily focused on neurodegenerative disorders, with companies that, such as Janssen actually being the exception, where they've still invested in mood disorders, and you'll hear from Wayne Drevitz later on in that regard. So this reduced investment, as Laura mentioned before, is for a number of factors. Next, please. It's unarguable that there's still a big need, an unmet medical need for new drugs to treat psychiatric disorders. So although the investment in drug discovery in this regard has decreased, there's still that patient need. Next slide, please. So this is the review of Mullard earlier this year, looking at the drug review, drug approvals, FDA drug approvals last year. There's 48 last year. The dark blue are the biologicals, so that's the antibodies, etc. And NMEs are the small molecules, the light blues. So you can see that there's 48 last year. Next, please. And you could just click on Laura, please. Yeah, so in this area of psychiatry, 2 of those 48 approvals were for psychiatric disorders, and that's actually 3 if you include esketamine. Esketamine wasn't formally included in that 48 because it's a new active ingredient rather than new chemical entity. But nevertheless, last year was a good year. On average, perhaps 1 or 2% of approvals in the psychiatric area. And so this, I think, highlights very nicely there's a paucity of investment in psychiatric drug discovery, despite the huge unmet need. And the 3 drugs I mentioned for psychiatry is the esketamine from Janssen, the lumateperone from Intracellular Therapeutics, and brexanolone from Sage, which I'll exemplify in the following few slides. Actually, that's not been approved for treatment-resistant depression. That's postpartum depression, so apologies for that mistake. Next slide, please. Go the next, please, Laura. And the next, please. Yeah, so this brexanolone is from Sage Therapeutics. It's a GABA receptor modulator approved, as I mentioned, for postpartum depression. Had a priority review and was achieved a breakthrough designation, which actually highlights several things, including the lack of existing drugs for treating postpartum depression and the willingness of the regulators, the FDA, to look quite favorably upon this compound. So what brexanolone is, it's a proprietary formulation of an endogenous neurosteroid, allopregnanolone. So allopregnanolone affects GABA receptor neurotransmission. It's an endogenous neurosteroid. And what Sage did was they took allopregnanolone and had a special formulation. And synthetic neurosteroids, so synthetic compounds are very similar to allopregnanolone, have been known for a long time to be anesthetic and anticonvulsant. Next slide, please. And this just shows the hypothesis. So it's been well described that following birth, mothers have a rapid drop of allopregnanolone. So next slide, please. Oh, sorry, next, click on, no, sorry. Could you go back, please, Laura? Yeah, so the hypothesis is, will an allopregnanolone replacement strategy have efficacy in postpartum depression? And these are the key clinical data that helped get brexanolone approved, changing Hamilton D-score in the brexanolone treated patients significantly greater than placebo. Next, please. So this was approved earlier last year, and that's despite the fact that brexanolone is a formulation of allopregnanolone requires a 2.5-day IV inpatient infusion. I've got serious side effects that merit the black box warning, which is shown there, and it costs about $34,000, so not the cheapest of medications. But that again highlights the willingness of regulators to consider molecules for where there is a very large unmet need. Next, please. And so going back to look at our low-hanging fruit of a GABA receptor positive allosteric modulator. Next, please. So this is essentially a benzodiazepine where we dial out the sedation, and we know that benzodiazepines have their effects at 4 subtypes of the GABA receptor, those with alpha 1, 2, 3, and 5. The structure is heteropentameric ion channel. Next slide, please. We know from some very elegant work from Hans Moll and colleagues in Zurich that the alpha 1 subtype is involved in sedation, alpha 2, 3 anxiolysis, and alpha 5 is cognition. Next slide, please. And this is a hypothesis Just looking at the panel on the right-hand side, if we can selectively modulate the alpha-2/3 receptors, so make the hole in the middle of the ion channel bigger selectively, we should have a non-sedating anxiolytic. Next, please. And so the challenge really is the medicinal chemistry one. How do we do that? And it is actually possible to selectively target those 2 of the 4 GABA receptors. Next slide, please. And that's exemplified by this compound from Pfizer. So this is their alpha-2/3 PAM. Clinically evaluated in pain unsuccessfully, but they did a very nice 7-patient study in photosensitive epilepsy where they showed a very marked effect at 17.5 and 52.5 milligrams. So that's the yellow triangles and red squares relative to placebo. So it's got a very clear target engagement and pharmacodynamic effect. And that compound was offloaded to a company called Cerevel when Pfizer got out of neuroscience. Next, please. So that's my summary. So yes, there are low-hanging fruit out there, and I think it's demonstrated by the repurposing of allopregnanolone for postpartum depression, and there's other potential in this area. So thank you.
Excellent. Thank you very much, John. There's I think we've probably got time for one question, and I was just interested to know, you've presented your portfolio, and given the kind of references we've had to sort of being patient-centric, I'm just wondering where those targets have come from and what kind of validation package you have before you'll actually pursue a target.
So we have to obviously go to the Medical Research Council or the Wellcome Trust or whoever and make a case that this is a valid drug target. And so what we have for some of those targets, like the AMPA receptor project and the GABA receptor project, is we have confidential in-house information from GSK for the AMPA receptor project, and some for the GABA project from AstraZeneca, who are collaborators on those projects. And they allow us to tell their story from confidential internal information, which backs up our proposal. So if you look at the literature, for example, you might think, well, GABA's been done, but we've got information from AstraZeneca, for example, that leads us to conclude actually it's not been done. There's still a significantly unfinished story there that we can exploit.